So that is, some people, they can't able to accelerate, even they can't able to ride the bike. Okay, so again, that also, it is behind within your DNA. It means that's what I'm telling, that your signature is written on your blood. Okay, so it means if you are healthy, your DNA is, good, your genes are good. If the DNA is, mutated, obviously you'll end with the disease. That the disease, maybe it is congenital or age-related diseases. Okay, so that's what I will make a strong statement. Your signature is written on your blood. Next slide. And that's what I mentioned in the office. So right now, the life expectancy of the of the people, the people living longer. So if you take 2 years, 2 decades ago, the life expectancy in India is a 58 or 60. So right now is 72 or 75. The women live longer than men. At least 5 years the women live longer. So like that. So 2 or 3 decades ago, the population of Tamil Nadu only 3 or 4 crores. Now it is double the amount. It's 8 crores. So the population is increasing, and then the aged population, especially in Tamil Nadu, more than 50, the population is very high than the younger population. So those people, definitely they'll prone for the disease after the age of 55 or 60. So again, why they are turning, I'm not talking about the infectious disease at all in this talk. I'm talking only the disease related to the genes. Okay, so if the gene is correct, definitely there is no disease. If the disease is already mutated, your signature is bad, you end with the disease, and then the disease express in the later stage, either at the age of 60 or 65. That's what, you are prone to develop, if your signature is bad, definitely you will prone to develop disease at the early stage. Next slide. So this is the right now the personalized medicine. Even the infectious disease, corona, so some people, they prone to develop, the mortality rate is there, some people not. We exposed, but they're prone to develop disease or death. So here, so once you know your own genome, definitely we can adopt a kind of medicine based on that. That is possible. It means, I will give an example. Suppose you have, your grandfathers or uncle, they may have a kind of disease, eye disease, either the cataract or glaucoma or diabetic retinopathy or age-related macular degeneration. So these are the age-related diseases. Once they turn into 45 or 50, you better to check your eye pressure, whether the pressure is very high or low. So that will affect your peripheral vision. If it is glaucoma, the elderly people, they can't able to see the periphery, they can able to see the center. Again, if it is a disease, the gene is mutated. Okay, because of the mutation, they have that kind of problem. So I think once you understand your own genome, you can postpone the onset of the disease. Suppose consider the onset of the disease at the age of 55, you can postpone the disease at least 60 or 65. One more information, you compare our population and then Western population. So in our population, the onset of the disease is 10 years, 1 decade before the onset of the disease when compared to the Western population. Consider one particular disease appearing at the age of 60, here it is 45 or 50. So there are so many reasons are there, even the UV radiation, most of the population is, agriculture based, they're exposing you to sunlight, that will cause a different kind of, factors, it is influence the disease. It may not be the right cause, but influence the disease. So like that, so that is the one more thing. Next slide. So I think here, what I would like to emphasize, so if you understand your own genome, okay, just simply get a blood, a 1 drop of blood or a 5 ml of blood, and then you can screen the entire genome of your particular genome. So there are the Human Genome Project is completed long back, 25 years ago. So now the number of genes, human gene is only 19,000. It means the human genome genes are 50,000 or 25,000 they told previously. Now the scenario is different. The human genome is shrinking. Only 19,000 genes are there, only 19,000. So if you pay like 40,000 rupees or $1,000, you can able to you know your own genome. Okay, so I think there is a reference sequence that is called HG19. Okay, they will refer, you consider, they will screen the entire genome of your DNA, and then they will compare with the normal DNA, the normal and your DNA. So where exactly the change happens in the 19,000 genes. So once you know, suppose consider, I told you, consider the eye diseases, glaucoma, there are some of the genes are there, like 500 genes are there. So if there is any mutation is there, what you can do, you can keep on checking your eye pressure. So whether that the disease is inherited from your father or your grandfather, that is the another catch. Suppose if there is any family history, like your father or grandfather having that particular disease, you have to educate yourself. I may prone to develop, because you already know the Mendelian genetics. Okay, the what is the you just the Mendelian experiment, tall and dwarf plant. If you cross that one, the capital T, small t, the heterozygous and homozygous. The first village generation, capital T, small t, that is heterozygous. Okay, so even the one defective copies, you already have that one. So in that scenario also, you prone to develop the disease. So you have to have it in mind. So I think I have a family history, and then I may prone to develop this particular disease after 50 or 60. I have to change my lifestyle to overcome that, or you can postpone the onset of the disease. But you have that kind of mental makeup. No, without slide I can able to talk. Don't worry. That your own genome, once you know your genome. So right now, that is called clinical NGS diagnostic. It is called, NGS, next generation sequencing. That's what we are routinely doing in the lab. So once you understand your own genome, even if a particular person is having the cancer, whether the drug is going to cure that particular cancer or not, that is depending upon your genome. For some people it will work that particular medicine. So based on the treatment, we can change the treatment. So that is very, very important to know your own genome. That is very important. You should not be panic. I have the disease, how to survive longer. Don't worry. So once you understand, you can change your lifestyle, and you can lead your life comfortably. Next slide. Okay, I think we have 46 chromosome. The mutation may occur at any kind of in the chromosome. Next slide. And then this is one slide. I think this is a very good representation. I will explain in detail. So as I mentioned, so okay. So here is a normal state and the normal state. In the very beginning introduction, I told you, if the gene is normal, there is no disease. He can able to live healthily. And if the gene is abnormal, see, you can see this particular thing. It is an induced signal. These are the induced signal. That genome to transcriptome and proteome. The induced signal, it may occur at any time. During the genome or in the transcriptome. Point. Okay. So I think these are the induced signal. Induced signal is nothing but a kind of mutation happen in that particular genome. So that's what So once you have the mutation, they will end with the altered state. It is a normal state and the normal state. Here the normal state converted into altered state. Altered state is nothing but a kind of disease. So this is a representation whether you are healthy or diseased. So once you understand your own personalized genome, you can understand what kind of disease you are prone for in future. Next slide. So I think because based on the topic, so this is one of the recent review from All India Institute of Medical Sciences. They compiled all the data, what is going on in India. So they compiled all the thing, and here they mentioned what is our contribution. Now I'm working for the past 23 years in Aravind. So they emphasized all the our research contribution in this review, and how you can take your research from bench to bedside. Next slide. Okay, so this is as I told you, is 19,000 genes. We have only 19,000 genes. Most of the in the genome, most of the DNA is very junk. It is not involved in the in the transcription or translation. So only 3% of them involved in the protein. It means the gene, it transcribe and translate, and then the protein, the end product is the protein. So that is the functional protein, and if there is any mutation, it is not going to form the right protein. So obviously end with the disease. So concerned with out of 19,000 genes, so there are approximately 547 genes are related to eye. Okay, so I think out of 19,000, we are majorly focusing on the either sometimes we'll focus on only that 550, or sometimes we will take the 19,000. The clinical exome, sometimes we'll take 6,000, 7,000 genes, and what are the associated genes? Not only the candidate genes, what are the associated genes for the disease? That is also we have to consider how it is modifier effect. Sometimes it may not directly associate to the disease. Some of the genes it reduce based on the environmental factors and other thing, so it is involved in the disease. So here I think approximately 547 genes are there in ocular diseases. Next slide. So I think this is central dogma. You already, know about what is the central dogma, how the transcription, translation, and the protein synthesis. Next slide. Next slide. Okay. So in addition, so you should not consider only the mitochondrial, your nuclear genome. So you have to consider the mitochondrial genome too. So here I think I would like to emphasize the mitochondrial genome. So there is mitochondria, it is having its own circular DNA, like a 16.5 kb. So if there is any mutation in the mitochondria, it will integrate to the next generation too. So and then the beauty is, it is not the, through them, it is the mitochondria, it is inherit through only mother, not from the father. That is the thing. Whatever the mitochondria we have, it is from our mother. Okay. So if there is any mitochondrial mutation, obviously there is one eye disease called Leber hereditary optic neuropathy. So if the mutation is there in the mitochondria, there are 3 mutations, and one of my students is here, he is working on that. What is the crosstalk between the nuclear genome and the mitochondrial genome? So, you had to what I'm emphasizing here, you have to concentrate not only the nuclear genome, you have to consider the mutation in the mitochondrial genome too. That is the message. Next slide. And see, this is the immunohistochemistry in the green color. It is not the nuclear genome. It is especially for the mitochondrial genome staining. Next slide. In a cell. Okay. So this is the age. Based on the age, you can get the disease, and we can classify the disease. Either it is a chromosomal disorder, or the single gene disorder, or the complex disorders. See the percentage here. See, the chromosomal disorder only point in decimals, but the 50% of them is complex disease. The aged people, all the aged people falls under this particular complex disease, because either the heart disease or glaucoma, everything falls under the diabetic retinopathy, everything falls under the 50%, and then age is the main factor. Once you get older, at least you're prone to develop this kind of disease. And then, as I told you, the 547 genes, sometimes a single gene, single disease, one particular gene, if it is mutated, that is associated with one particular disease. But sometimes, it's a complex disease. There are more than 30 or 40 genes, or 20 genes is the candidate gene for one particular disease. So those are, complex as well as the environmental factors. For example, the cataract, our temperature is very high, 40, 45, sometimes 50 degrees centigrade, and at the summer. So if we expose to that one, obviously they will get the cataract, and even your gene is mutated, and especially the women, they're prone to develop the cataract early than men. The reason because after the menopause, there are a lot of changes in the body, as well as they're exposed to smoke, because when they cook, they're exposed to smoke. That will trigger the onset of the disease. Okay, so those comes under the complex disorders. So based on this, we can classify it is a chromosomal disorder or a single gene disorder, and then complex disorder. So for the past 25 years, we extensively studied all the 3 categories of the eye diseases. Next slide. See, these are the mode of inheritance. Some disease, it inherit to the father to son, that is called autosomal dominant. Sometimes it skips from one generation to another, that is called recessive. You can see here, this is the mitochondrial disorder, as I mentioned, is maternally inherited. See here, the father, I think this particular children is not affected, but others are unaffected. All the mitochondrial DNA, whatever we are mitochondria, we are getting from only from mother, not from father. This is kind of mode of inheritance. Okay, so if the consanguineous marriages, if the consanguineous marriages is there, definitely the recessive disease is the chance is very high. Okay, so if it is autosomal dominant disease, the disease is a 50-50 chance, and then if it is a recessive disease, it's a 25% chance. But the severity of the disease is very high in recessive disease. That's what people are recommending, avoid consanguineous marriages. Next slide. Okay, I think in the mitochondria, so that's what I mentioned with Andrew, there is a poster, So through mitochondria, what you can do, so people studied how the human migrated from Africa to other countries. Okay, so through mitochondria, we are also working on that. It's called haplogroup. We all South Indians, they comes under M haplogroup. So we are M haplogroup. So based on that, how do you find? You can ask a question. How do you find out the haplogroup? So you can screen the entire mitochondrial genome, like a 16.5 kb, entirely screen, and then one particular marker is defective or mutated for you and me. So based on that, we can track. So the entire population is mutated. They will clump together in one particular I group that is called M haplogroup. Next slide. Okay, so this is I think I don't want to go in technical details. So right now we are adopting the next generation sequencing. We are not focusing on one particular gene. We are screening the entire genome. As I told you, either the 19,000 genes or the 600 genes. So you can go for the whole exome sequencing or genome sequencing, and then you can find out the exact variation in the patient and then the family members. That's what we are adopting. Next slide. And then see here, so this is the patient, and these are the technologies, high-end technologies, not only in genome. So you can deal with the transcriptomics, RNA. No, that is also you can do, and then epigenetics. Epigenetics in the sense, there is no change in your DNA, but definitely there is but people getting disease. It means the histone modification. So those things, that is called the CpG islands. The entire genome, if you screen, so what kind of CpG island is, giving the track for the particular disease. So those things, these are the DNA analysis. We have our own pipeline to analyze the data. Once you get the entire data, so the bioinformatic analysis, we find out, so this particular patient is having this particular mutation. This is very pathogenic, and that's what he got the disease. So like that, so we can come to a conclusion. So, and then we can recommend whether this particular mutation is going to inherit to the next generation. Suppose consider in your family, father and mother and then affected child. So they want to have a new baby, and in that scenario, you can trios. You can screen all the father, mother, and the affected child, and what is the chance to get the next baby, whether it is going to be normal or abnormal. That is possible through this NGS. Next slide. Okay, I think bioinformatics, I don't know the fine bioinformatics team is here or not, but that is a very, very challenging one. So you have to understand perfectly, then only, even if you have the raw data, so you have to come up with the right information, then only you can be able to give the right information to the patient. Next slide. Okay, so then we'll come to the eye. So this is the structure of the eye. You already studied in the 10th grade. So this is the structure of the eye. So this is the cornea, and this is the optic now. This is the entire structure of the eye. So you can name it. You may think it's nothing, because his eye is a small organ. Nothing is there. You may think because you are normal, your sight is good, but concern with the prevalence of the eye disease is very, very high. Especially in India and other countries, the people having many kind of disorders in eye. Suppose if there is any problem in the cornea, so the cornea is people are donating the eye, right? After death, people are donating the eye. We are taking only the cornea. This is the region we are transplanting to the patient. Okay, and if there is any the compatibility. Okay, so even the single layer cornea, the cornea is epithelium, stroma, and endothelium. So each and every layer, if it is defective, if the gene is defective, it may not form the right cornea or endothelium. Okay, because the gene is defective, that's what during the development it won't form the right layer. It means if you see the developmental biology, one particular gene is mutated. Consider one particular gene is mutated. Okay, it's a neurocrust origin like that. So during the development, that particular cell, it will migrate to the globe. Once it is migrated to the globe, it will form the right structure. The cornea, iris, lens. That is because it is migrating, and then the gene is normal. It will forming the right eye. Suppose anophthalmos. Sometimes people born with no eyes, or microphthalmos, small eye like that. There are so many complications because the gene is defective. Once the gene is defective, so the migration of the cells, during the development, it won't form the right structure. Okay, so that is the problem. Then you can't be able to get the right visual acuity. Then that is a major complication. So here, like that, we extensively studied for the past 23 years, cataract, age-related macular degeneration, liberated optic neuropathy, like that, glaucoma, and cornea. So maybe I will tell you, explain in detail about cornea. Next slide. So I think these are I have 20 PhD students already got awarded. I think each one, I can give them a particular problem, either the gene screening or how that particular gene is involved in the pathogenesis, like that, Now we are well documented this in our population, like in Southern Indian population, how many patients are affected, or the spectrum of genetic variation in the population is possible. Suppose consider like a diabetic retinopathy, like these are the genes, So if you go and search, yes, this particular gene is prone to develop in a particular population, like that, is well documented all the diseases or the genes. Next slide. These are the list. Next slide. Okay, so I think Andrew mentioned about the diabetic retinopathy. Maybe I will briefly explain diabetic retinopathy. So that there are two kind of, forget about the type 1 diabetes, type 2 diabetes, because after the age of 45, they may get them with diabetes, uncontrol of the hyperglycemic, people get diabetic. So some people, they are diabetic, but they never develop the retinopathy. Retinopathy in the sense, so these are the vessels, it will come in the eye. If you are not maintaining the sugar level, but if it is hyperglycemic, there is chance to burst the vessels, and then it will leak the blood, and then suddenly it lose the vision. So one group of people, they will develop retinopathy. One group, they never, even though they are diabetic, they never develop the retinopathy. So it means, so we are in many scientists, they are working. So what is the difference between that particular diabetic group and the diabetic retinopathy group? Okay, so they are working in gene level, protein level, in like other microRNA, like that, they are extensively working on that. So here, so what one of my students right now, she is in California. So I think she worked on this particular thing. We have collected more than 500 samples of the diabetic and diabetic retinopathy, and we did the next generation sequencing, and then we identified some of the kind of marker, So that marker is prone to develop instead of diabetic retinopathy. So this kind of studies we already developed through diabetic retinopathy. In the sense, what I'm trying to say is, so I think suppose consider a person is diabetic, better to control his normal sugar, or if it is and then they never come to eye hospital. If it is diabetic, they never care about the eye. They will check only the blood sugar, control the blood sugar, but there is chance to affect the eye. Okay, so they have to come to the eye hospital. Next slide. So this is our paper as the Sugandha Lakshmi. Right now she is in California. She is that is her PhD work. The entire PhD work is we published in BMC, Medical Genetics. Next slide. Okay, so I think as I mentioned, the cornea. So this is epithelium, stroma, and endothelium. Each one, it is having its own gene. If the gene is mutated to that particular patient, obviously there is abnormality in that particular epithelium and endothelium. The thing is, see here, this is multi-layered, and this is only single layered. It's only single layer. So this is very difficult to regenerate. You can compare with the nerve cell. If there is any wound in this part, it will heal. But if it is in the nerves tissue, it will take long time. So the same thing here, it won't regenerate. If there is if the cell is dead, it is never regenerate. Okay. So in 2006, till 2006, even though the Human Genome Project is completed, nobody knows what is the candidate gene responsible for this particular disease or this particular layer. Nobody knows. Okay. So fortunately, I think Aravind, I think the high volume, we are getting a lot of patients. We are seeing 2,000 patients per day. So I think the consanguineous marriage is very high in Southern Indian population, and well established ophthalmologists are there. We can able to recruit the right family to study this kind of genetic study. So here, I think in 2006, only 5 families identified all over the world. I think 7, 3 from Aravind, 5 from Aravind, 2 from Singapore, or in Myanmar. Okay, so like that, we pooled all the data, and then we did the linkage analysis, and we identified what is the candidate gene for this particular disease. The disease candidate gene is present in the 20th chromosome. Through the high-end sequencing, we identified, yes, this particular disease is responsible for this particular disease. So here, the inheritance, as I mentioned, it is autosomal recessive. So it means grandfather have the disease, but his son may not have the disease, but his grandson having the disease. So based on this, so we linkage analysis, and then we identified the gene that is responsible for this particular disease. That's what, we published this paper in Nature Genetics. I'm one of the first author in that paper. So I think, this is very, very important. What I'm trying to say is, you better to, again, I would like to emphasize, you better to understand your own genome. That will help you for your disease-free, to lead a disease-free life. Next slide. So I think this is the patient. See here, the endothelium is not that much. See, the endothelium, entire endothelium is gone for this particular patient. Okay. So because the gene is defective. Next slide. So this is the candidate gene, the SLC4A11 gene. This is present in the 20th chromosome. So when we screen the entire gene, so these are the mutations we identified, and we have to functionally prove. Suppose like one particular mutation is happened, how it is functionally it is associated with the disease. Next slide. So we cloned that particular gene, we made the site-directed mutagenesis, and then see here the wild type is like that. If it is mutated, see the pattern, is entirely different. So it means it is not secreting the right protein, it is truncated. So that's what people end with the disease. It means, I think you have to understand what is haploinsufficiency. Okay, suppose the gene it will produce the right protein that is for the function of the one particular thing. Suppose saliva, secretion of saliva. For some people, even they can't able to secrete saliva. Okay, so it means the gene is normal. If the mutation is there, the problem is the protein. Protein is not. If it is truncated, it is not doing the perfect job of the function of the particular gene. Okay, so here through Western blot analysis, we proved, yes, this particular mutation is associated with the disease. Next slide. And then obviously immunohistochemistry, you have to understand the protein synthesis. Some of the protein will come to the cell surface. It will synthesize, it will come to the cell surface, or sometimes it will come into the cytoplasm. Okay, in the cytoplasm. It means the protein is extracellular or intracellular. Okay, so when you grow the cells in the medium, if it is come out of the cells, it is the extracellular, and then if it is sitting inside the cell, it's intracellular. So through that, you can understand. Obviously, the mutation is there. Once it is truncated, the protein it stay inside the cell. So through the immunohistochemistry, so what is the difference between the mutation and then the normal and then the wild type? So based on this, the immunohistochemistry, we confirmed, yes, this particular mutation is associated with the disease. Next slide. And this is the paper we published in Nature Genetics. Next slide. I think this is one of the very interesting family. So when we submitted this paper to Human Genetics, you can't be able to mimic this kind of experiment in an animal model. Okay. So see here. So see the pedigree. So this man, he married both the elder sister and younger sister, and then the younger sister, there is no issue, and here she is having three sons, one is affected and the another two are carrier. You can see here, this is the younger sister and then the elder sister. The elder sister having the babies, they are normal. This guy, is affected. Okay, so this is a kind of, here the interesting thing is, this is one particular gene that is called FOXL2, FOXL2. So if I think you know the structure of the gene, exon and introns and everything. So here there is no introns, only one exon, the entire gene, and then in one particular place, there is a lot of alanine repeats. The alanine amino acid, it will repeat 18 times. You and me having the same sequence in that particular gene, FOXL2, alanine, like 18 times. If there is any one more alanine is added, definitely you will end with this kind of disorder. Okay, so we carefully, it means here this particular protein, some of the proteins, it will come, as I told you, it has come to the surface. Some of the proteins it will accumulate only in the nucleus. Next slide, please. So here, once you understand where exactly the protein is sitting, so here we confirmed this mutated protein is not coming to the nucleus. It is sitting only in the cytoplasm. So through the immunohistochemistry, we confirmed, yes, this is the culprit for that particular patient. Okay, next slide. And another, so our research, the aim is, so you are doing the bench work, how to transfer your work to the bedside, because the patient should get benefited. Okay, so this is albinism. Please don't confuse the leukoderma. So this is albinism. There is because our skin is dark, and then because the tyrosine, TYR is good, because we can, even if we have high radiation because of the melanin, we can able to withstand. So in Australia, people, they will go for sunbathing in the seashore, they will apply oil and everything, but for us, there is no point to apply oil, but we have the melanin because our skin is dark. Okay, so here there is a one particular gene. Next slide, please. So this is the tyrosine TYR and eumelanin. This is the pathway. Please, recall the biochemistry, like a glycolysis to pyruvate. The same pathway here, tyrosine to eumelanin. If there is any mutation, like either here or here, there are so many 5 genes are there. So is there any mutation happens in this particular pathway, they will end with the particular disease. Okay, again it is autosomal recessive disease. Next slide. Is the same story, the TYR gene, where the mutations are there. Next slide. And this is, now that's what I'm telling you, you have to transfer your research from bench to bedside. So this is another family. They want to, have a normal child. The father is normal, mother is normal, and then they have an affected child. See here. Here, okay. So the mother, father, and then the first baby, is aborted, and this baby is, already having the disease. See here, it's a heterozygous condition. Because of these alleles, he got it, that's what he's affected. And another baby, so they want to have a second baby, and they came to us, and here the problem is, before conceive, within a short period of time, we are supposed to get the amniotic fluid, and then we know the entire sequence of the father and mother. Okay, we know exactly what is happened in the TYR gene, and we know the genotyping where exactly the mutation, and because this baby is already affected, and then we got, once that the mother is conceived for a second time, so we got the amniotic fluid from, they obviously in eye hospital you can't able to take the amniotic fluid. They had to go to Chennai, and we got the sample from Chennai, and we isolated the DNA, and then we identified that particular DNA. There is no change exactly like a mother or affected child. So what we informed to the patient, see, whatever the variation in your DNA, it is not in the amniotic fluid. Okay, in the amniotic fluid, the chances, he may be normal. But now we, as a genetic counselor, you should not advise, okay, go ahead, you can have the baby. Something you should not do that. You have to tell the scientific truth. Yes, mother is having the variation, father is having this variation, the affected child is having the variation, the chance is like this for the next baby. Okay, whether you want to continue the pregnancy or you want to abort, the decision is yours. You know, you can take your own decision, and then you know you will say, you know, my language is the ball is in your court. You know, you can play. You know, it's up to you. Okay, so like that, you know, we can able to transfer whatever the research you are doing, you know, to be translated to the bedside. Next slide. I think forget about this. This is one particular community. You know, I don't want to reveal the community. If they have that kind, even if they come to us, I can able to say, I think definitely you have this kind of problem. Okay. So next slide. It's called care diagnosis. So before marriage, we can able to sequence the entire genome, and be able to say, so this is the variation, and why not you have the next partner, something like that. No, that is possible. You know, that is we can able to say. Next slide. Okay, this is how to take the amniotic fluid. Next slide. Next slide. I think I don't do good. Then the entire next slide. Then they came to us. You know, they got the normal baby. They are very happy, and then they want to check the entire family. They came to us, you know, and they want to know their status. You know, what I want to know my own genotype, and then we screened the entire family, and then we told whether you are having the homozygous, heterozygous compound, like that, you know, what is the future, how to choose your life partner, like that. No, that is possible. Okay, next slide. Okay, I think how long I can take further? Okay, okay. So now I will switch the gear to the gene therapy, you know. Okay, so once you have your own, again I will summarize. Suppose you better to check your own genome to postpone the onset of the disease. Very simple. I can put it in a different way. There is a Centurion project. Centurion project in the sense, those who lived 100 years, especially if you go to Tokyo, in Tokyo itself, there are more than 200 people are living more than 100 years. Okay, you can collect only their DNA, and then you can sequence the entire genome of their DNA, and then find out what is the gene responsible for the longevity of the life. Okay, so why they are living longer when compared to the normal the other people. Okay, so I think so they identified, yes, some of the genes, like 7 genes are associated with the longevity of the life, and then those genes, you know, again it is correlated with the age onset. Those who are having suppose one particular disease, they will get the disease at the age of 70. You know, in America it is called age-related macular degeneration. So the age-related onset disease also correlated with the longevity of the life, those genes. Okay, so like that. So the sincere advice is you understand your own genome, and then because your signature is written on your blood. Okay, so now gene therapy, you know, like those who are failed with the chemotherapy, radiation therapy. Okay, so consider cancer, anything, you know. So I think they will treat with the chemotherapy or radiation therapy, and then if there is failure in radiation and the chemotherapy, what is the alternative therapy? Alternative therapy is, I mean, the viral therapy. Okay, gene therapy. So here, I think I don't want to go in detail. Maybe I can take a separate class. Next slide. Next slide. So next slide. That is the definition for the next slide. Okay. So I think gene therapy, in the sense, consider one particular gene is defective. That's what, you know, you are into with this particular disease. If we alter that particular gene through the viral vectors, we can you know, that particular gene, it will secrete the right protein, and then they will get, I mean, improvement in the disease. So that is the concept. So you are supposed to transfer the normal gene to the, suppose consider one patient is a defective gene. Okay, so you are supposed to remove that particular gene, and then you have to transfer the normal gene through viral vectors. So these are the viral vectors: HSV1, herpes simplex virus, adenovirus, adeno-associated virus, retroviruses. And then these are the non-viral vectors. It may be a naked DNA or liposomes or molecular conjugates. I'm not going to talk about this. So this is the, I mean, I will take, I mean, only the viral DNA. So HSV1 is nothing but the herpes simplex virus. The genome of, as I told you, we have 19,000 genes. The HSV1 is a 152 kb, is a very big gene. Again, you have to understand what is the genetic material of the virus. You know, some virus, it is a single stranded RNA, double stranded RNA, some of them are single stranded DNA RNA, like that, you know, there are a lot of genetic material is different. So here the HSV1 is a double stranded DNA virus, and then the advantage between the HSV1 and the adenovirus, you know, this genome is very high and this genome is very, very small. So you can accommodate here, I mean, your gene of interest comfortably, and here I think is a problem because in the cloning strategy. So here HSV1, the herpes simplex virus, and then I think immunology. So you know, the virus, you know, is a replicate through the peripheral nervous system. You know, it will infect through, it go to the it infect to the periphery, and then it will go to the central nervous system, and then it will start to replicate. It means it is called a neurotraffic. It will replicate only in the neuron. Okay, some of the virus it will replicate only in the lymphocytes. Okay, so lymphocytes, lymphotraffic, and then the neurotraffic. So this virus is a neurotraffic, it replicate only in the neuron through the peripheral nervous system, to the central nervous system. Then central nervous system, it will go to the brain, and then it will form the encephalitis, and then go for the mortality. That's the end stage is the mortality. Okay, so the adenovirus, very recently they celebrated the 50th year of the adenovirus and adeno-associated virus. They identified in the very beginning only adenovirus. Okay, this particular genome, the adeno, why they called it is adeno-associated virus. Consider this is the adenovirus genome. The adeno-associated virus genome is integrated into the adenovirus, but they can't able to separate in the very beginning. You know, they thought, you know, is only one genome. Then through high-end sequencing, they can able to separate two genomes. That is, one is adenovirus and then adeno-associated virus. They given a different name. So if you take this particular virus, you know, this is very, very safety when compared to the HSV1, because the safety is very important, because you know immunology, the antigen antibody, and then it will go into the system. It should not affect your system. Okay, the HSV, the adeno-associated virus, it is not harm to the human being. Okay, so it means, and then the replication. So here the HSV1, consider the virus, it will infect to one cell, and then it will replicate, and then the progeny, you know, it will go to the next cell. It is called cell-to-cell spread. You know, it will spread quickly. But the concern with the adeno-associated virus, one virus, one cell. That's it. You know, it is never, you know, there is no cell-to-cell spread. So if you adopt this kind of a virus, that is good for gene therapy. Okay. So you can read paper right now. They will use either adeno-associated virus or lentiviruses for gene therapy. It means you take the adeno-associated virus, lentivirus, and then you can clone the gene of interest in the lentivirus or associated virus, and then you take the clinical grade virus and then put into the right organ for how to rectify the defective gene. That is the strategy. Okay, next slide. Okay, okay, I think that is the viral genome. I think I don't know. Okay, so this is the viral genome. This is HSV1. Next slide. This is the electron microscope photograph. Next slide. See, this is I think if you are interested in immunology, is a very, very, very interesting thing. You know, see here the virus is very clever. Like the COVID, you know, the SARS virus and COVID virus also the same. I mean, family. So here, how the virus, you know, see here, this box and this box, you know, it is exactly the same sequence. Okay, this is called a kind of same repeat, and this box and this box, you know, exactly the same sequence. This is called unique long and unique short. No, this is the entire genome of that particular virus. These are the functional genes. Suppose the GB, GL, these are the replicative genes. So the viral replication is very, very important. You know, then only it can be able to cell-to-cell spread. So if we knock out the replicative genes, you know, it won't be replicated, right? Okay, the replication, you know, is the first stage to stop the cell-to-cell spread. So if you target the replicative genes, so obviously you can make it a recombinant virus. So that is the strategy. Either you can choose the replicative genes or some of the genes, you know, in the unique short region. So it means if you have the double knockout, in one region here and one region here, it is may not be a pathogenic. It is, it is carrying only your gene of interest, and then you can easily transfer to the patient. So that is the concept. It means you are making a recombinant virus that is not, should not behave like a wild type. That is the thing. Suppose if you put this particular virus into the mice, immediately, within 24 hours, 48 hours, it will die. As I mentioned, through the foot fat is the peripheral nervous system. It will go to the central nervous system, and then it will form the encephalitis. So that is the mode of transmission from PNS to CNS. So if you knock out those genes, what I told you, the replicated genes here and there, you are that is a very simple, it is not virulent and non-virulent. Very simple terminology. Virulent, non-virulent. You can make the virus as a non-virulent, and then you can order the virus, okay, carry this particular gene, and then you can transport to the particular organ. That is the concept. Next slide. Okay, next slide. Okay, another thing is, this is a very good, strategy concerned with immunology. See here, consider this is a cell, human cell. The virus infect to the cell, it will replicate, and then the proteins, the viral protein express, please listen, that the viral protein express in the infected cell surface of the human as well as in the viral envelope. Okay, so it will express both in the cell surface of the human cells as well as in the envelope. It means those proteins act as a receptor. Okay. So here, this is the, see, as I told you, GD and, see, consider this is the one particular cell, this is the receptor, and then the IgG. This is the IgG. So this receptor is, the FAB end is, binding to a specifically binding to one particular domain, the FAB end is free. Okay. So the FAB end, it should bind to the next other receptor. Okay. That is the mechanism how the virus should escape from the immune attack. Even though your antibody is good, the virus, it is having its own mechanism to escape from the immune attack. Okay, understand? See, two receptors, because the virus is already infected to the cell, and then it will form and the receptors are expressing in the cell surface of the human as well as in the virus. So it is having two receptors. The antibody, the FAB and the FC end, it will bind in one end, this end will bind to another protein, another receptor. So because of this mechanism, this is called bipolar bridging. The virus, it is forming a bipolar bridging to escape from the immune attack. Obviously, it should live, because that's what, the survival of the fittest, right? So survival of the fittest, it should live for its survival. So to escape from the immune attack, this particular virus, doing the strategy. So our strategy is, so if we knock out this particular region, where the FAB end or FC end, if you knock out those, region, you can make a strategy. Okay, so it means even though the virus, it can't able to bind, it will die. Okay, so that kind of strategy, as I mentioned, either you have to knock out the replicated genes or associated genes, or this kind of strategy. If it's so, you can make a virulent virus instead of the virulent virus. Next slide. So next slide. So this is how the virus, once you put the virus, how it is expressing, that kind of thing. Next slide is immunohistochemistry, where the domain and everything. Next slide. Next slide. Okay, next slide. I think, next slide. Next slide, these are the, okay. Okay, so this is the virus, as I mentioned. So the wild type, it will form immediately, death, because it is a virulent. But we made a recombinant virus that is called G207. G represents the Georgetown, because we worked in the Lombardi Cancer Center, the Georgetown. So here, I think this particular virus, whether it is having the clinical grade, because you can't able to take a virus from the laboratory and then put it in the brain. That is not the right way. You have to make it a clinical grade virus. Clinical grade in the sense, even the foreign material or the antigen enter into the system, it should not make any difference. Okay, so we given this particular virus to a Canadian company, and then they made the clinical grade virus, and then you take that virus. Next slide. Next slide. See, these are the knockout. Next slide. Okay, next slide. Next slide. Okay, maybe here, see here, this is before going to the human, you're supposed to prove whether it is working in the mouse model or not. So this is the tumor. So you take the nude mouse, because by the nude mouse there is no T cells. You take the cancerous cell, especially the glioma, because we are worked on the glioma and glioblastoma, the brain tumor. You take some 1 million cells, 10 to the power 6 cells. You take that cells and you could put in the flank. Okay, in the flank, in the sense you should not inject into the bone or in the muscle. You have to accommodate the cells between the space between the skin and then muscle in the rib. Okay, so if you accommodate within a short span of time, it will develop a tumor. This is the entire the tumor mass of the glioma. Then you have to inject the virus, the recombinant virus. Okay, if you put the virulent virus, it will die. But this is our target. So if you put the recombinant virus, see this region. So these regions, this virus, it is go infect the viral the cancerous cell, and then it will go to cell-to-cell spread. So cell-to-cell spread, it is not 100%, killing. It will killing only like a 50 to 60% of the tumor mass. So these are nothing but kind of staining, because when you clone, you have to put a marker. If you stain with the laxi, see, this is the tumor mass, a kind of necrosis. These are the killed cells. The debris is there. It means when you measure the tumor volume of the mouse, like 2 or 3 weeks, the tumor volume is reduced. Okay, so it means when you take the same thing to the patient, keep on taking the MRI, and then you can see that the regression of the tumor in the brain or in the mouse model. That is the concept. So we proved in the mouse model. Next slide. And then this is a this is a kind of a kind of strategy, like take the tumor cells, and then exactly how you are going to put it in the human. So you have to make a kind of a stereotactic injection, anesthetize the animal, and then you can just make a borehole in the particular skull, and then you can make through the Hamilton syringe. It means you take through a kind of setup, you deliver that particular cells into the tumor, like go 3 millimeter down the brain and then come up 1 millimeter, and then you can accommodate within the 2 millimeters the cells, and then how we are mimicking exactly the same thing in the human. So like that, we proved in the mouse model as well as in the monkey model how the tumor, growing in the brain. Then once the growth is there, then you can inject the virus, because that is our target. Those who are failed with the chemotherapy, radiation therapy, put the alternative therapies, the viral therapy, inject the virus in the same way, and then that is the structure I showed you, the tumor, how the tumor volume is reduced. So here the message is, once a patient is affected by cancer, please listen. Once a patient is having the cancer, it is impossible to cure the cancer, impossible. Even a single cell is sufficient to proliferate, and again it will metastatic to the rest of the organ. Okay, right? So here what you can do, you adopt the chemotherapy, radiation therapy, like alternative therapy, viral therapy. You can prolong the life expectancy of the patient. Suppose consider he is going to die within 6 months, but you can prolong the life expectancy of the particular patient for another 3 or 4 years. If you have the right medicine or Medicare or your insurance, definitely you can postpone the onset of the disease. Okay, next slide. So we adopted the same kind of strategy. Next slide. Next slide. Next slide. I think if you are really interested, you can simply type my name, and in the year 2000, this paper is published in Journal of Virology. Okay, so so the concept is, so we recruited the patients in Washington. See, this is the patient. So next slide. Next slide. Go to the MRI. Next slide. Back. Yes. The MRI, please. So in the sense, you have to locate where exactly the tumor is, whether here or there. Based on that, you have to make the position. Then only you can able to inject the virus. So you just make a MRI scan, and then inject the virus, and then make a seal it, and then so let him you can observe the pre and post op, or pre and post treatment, and then how the tumor is regressing. So that way, so our result is, obviously this virus, because you are putting the virus into the system, this virus is not harm to the human being, so it is helping to reduce the tumor volume of the patient, especially in the glioma. So I think the happily they are living longer for another 3 or 4 years. There is no side effect because of the based on this viral therapy. Okay, see, okay, I think okay, so I think this is the work. We transfer that same technology to ophthalmology. You can ask a question, how whatever we learned, we have to transfer that particular thing to the ophthalmology. So in ophthalmology, so there is a, please, if you are really interested, you can type RP65, retinal pigment epithelium 65. That is the name of the gene. Okay, the gene therapy is possible for, okay, forget about that, please. The gene therapy is possible for some of the eye disease. Possible. Okay, so now right now people are working on with many eye diseases in the mouse model, and then the successful gene therapy model eye disease is approved by the US government, that is called FDA. Before you had, when you getting the approval from the FDA, is a lot of procedures there, like whether you have to prove this is not having any side effects, like that, the pathologically everything, you have to prove yes there is no effect. So the same way there is a RP65. I'll come to the point. RP65. Okay, please, if you are interested in the lunchtime, you can simply type RP65 gene therapy. Okay, so the RP65 gene therapy is nothing but a gene responsible for vitamin A metabolism. So people are telling you take multivitamins, vitamin A, that is good for your health, good for your eyes. If you have, if you consume this vitamin A, you will get bright, eyesight, like that. The reason, because I think the RP65, it means your gene RP65 is normal. Okay, your gene RP65 is normal. That's whatever you are consuming, the fruits or thing, that is digested, and digested those micronutrients is taking to the retina, and then it is nourishing the cell. Okay, right? So if there is a defect in the RP65, even consider a particular patient is having the mutation in the RP65, even that the patient is consuming healthy food, an apple for a day, keep the doctor away, I think that is not going to helpful for that particular patient. Okay, right? So I think because the gene is defective, it is the micronutrient not able to take him to the nourish the cell. Okay, so I think this RP65, it is FDA approved. First, they got the approval in a large animal model. They used the dog model. Even you can type that, that the particular dog, they took the picture in front of the White House. Okay, and then because even the senators, they approved for this particular trial. Okay, so first, they proved it in the animal model. Yes, this particular gene therapy is possible. They proved it in on a dog model. Okay, so in the sense, right? So consider the patient is having a mutation in the RP65. So he is having a defective vision. Okay, so I think what you have to do, you have to take the blood, screen it, and then find out the mutation. Yes, this particular patient is, தம்பி ஒரு ஃபோல்டர்லேயும் இருக்கும், ஒரு இதுலேயும் இதுதான் அதான். ஒரு திருக்குறள் இருக்குல்ல, ஐ திங்க் ஸோ கல்வியை திருட முடியாது. ஈவன் ஐ டோன்ட் நோட் டு டிபெண்ட் ஆன் பிபிடி. ஸோ everything is within you, I think you can deliver even at space. Okay, so okay, so this is the MRI. Okay, so this is the stereotactic injection of the particular patient, and I think there co-workers, Dr. Robert Matusa, and they are the famous surgeon in Boston. You know, the Boston, that's a well-known medical school, Boston. So they are right now, they're the chief in the Department of Neurosurgery. Okay, this is my paper. And okay, we'll come to that, the gene therapy for RP65. Okay, so this is, that is, okay, so this is called a LCA, Leber congenital, whatever I told you, that is called a Leber congenital amaurosis. This is a name of the disease, LCA. You can simply see what I will do. If I'm in your position, what I will do, I never take points. I never take points. No one is having the pad, no. You please use my pad and pencil during the lecture, you know. I think I never take any points. I will take only the key words, you know. Like LCA, I think that is more than sufficient to pull down the information from the network, right? So no need to write, you know. Yeah, kind of, I mean, what is called short and writing, you know. Please, at least you have that kind of, at least you practice yourself, you know. And then the human genome. What is the normal human genome? I told you some word. HG19. You know. Okay. Okay. So I think you guys are, I mean, hungry. So the LCA, Leber congenital amaurosis. This is one of the disease. See here, this patient. This patient, you know, you may think, you know, it is a very rare, is a rare disease, but concerned with the population, you know, the population, the huge population. You know, in India, even if it is 1 in 30,000, 150,000, this kind of patient is, I mean, we can able to identify, they will come for the treatment to the hospital. Okay, so this particular patient, you know, they will keep on poking the eyes, you know, like, okay, so there is a photophobic, they can't able to see the bright light, you know. Obviously, there is a defect in the RP65 gene. And the treatment is, as I mentioned, they already proved in their dog model, and they want to transfer that kind of gene therapy to the human. Okay, so what we did, I think 2 or 3 paper we published, and then 2 PhDs are currently working on that. And so, okay, so this is the eye. So you can as I mentioned, the HSV1 or adenovirus virus. You can take that virus and then you can incorporate through the cloning strategy the RP65 gene, the normal gene, and then these are the, I mean, that the expression. You can take that virus, you can put it exactly in the subretinal space. As I mentioned, in the tumor, you have to put it in the in the where exactly the glioma, glioblastoma there, no, because of the scanning. But here, the problem here, the LCA is a retinal disease. You have to accommodate the virus with the corrected gene into that subretinal space. Okay, so it's a very tiny amount. You can inject the subretinally, and then after some time, you know, you can observe the patient, the before and after the visual acuity. Suppose consider the patient is coming today. He is having the defect. Ask him to, you know, you can arrange the table and chair in zigzag manner, but he do, because he is not having the vision. But if he walk through, he can't able to go from that end to this end because he will bump. Okay, because he is saying the defective, right? To prove that one, so you can accommodate, you can virus, you know, into the subretinal space of the patient. Wait for some time, 2 or 3 months, and then you do the same kind of experiment before and after surgery, accommodating this virus. He can able to pass through because the visual acuity may be increased. You know, he can't able to bump because the vision is improved. Understand? The vision, see, that is called, what I'm trying to say is, consider a particular patient is, blind. Okay? So, if he get a vision, hand movement, that is great achievement. Yes or no? Right? For everything, you know, he's supposed to depend on others to go here and there. But even if he get a hand movement, okay, so I'm here, okay, in that, you know, that is very, very excellent thing. So they conducted that kind of experiment in the animal model as well as in the human. They will the same way they will arrange a chair. You will ask him to come through after the treatment. After some time, you know, if the virus work out, he will pass through in a particular time. In the same time, they will reduce the light, that is called lux, in a high lux and low lux, because you can't able to, sometimes, the light is there or not. Okay, so they will diminish, and then they will increase the light, and then different lux, they will ask the patient to come through this way, and they will assess whether the visual acuity is improved or not. Okay, so you can ask a question, what is going on in Aravind Hospital? Okay, so what we did, so we went to the blind school, and there, simply here, the patients are very poor and ignorant. They will say something, you are not going to get vision, So what we did, we went to the blind school, we screened those patients, and whoever is positive for RP65, so we educated them, like out of 100 patients, not all the patients are positive for this particular mutation, only 8 patients. We communicated through, the Government of India. Right now, the ICMR, Indian Council of Medical Research, they formed a committee that is called gene therapy trials. Okay, so right now recently they started. Right now they are making a clinical grade virus. Please, you remember I told you we did it in Canada. The same way, to develop a clinical grade virus right now in Narayan Nitralaya, Bangalore, so I'm in the committee. So we are making a clinical grade virus. You take that virus if the patient is willing, with the approval of the Government of India and Indian Council of Medical Research, we can able to treat these kind of patients. But still the government is not approved, but we are in the process to at least identify the patients, they are eligible for that particular therapy. So once it is get it done, after the approval, they are eligible to go for therapy. Okay, next slide. Next slide. I think I will wind up now, right? Okay, next slide. Oh, okay, I will do it myself. Okay, I think maybe this is the advice is very, very, very easy to everyone. Okay, I think this is the very right time. You already, house arrest. Now you are free now. You are moving here and there. I think that's what Andrew told. You are very good in college. Very good atmosphere. Please think about that. Don't you have to be very, very careful. It's a very, very competitive world. You enjoy as well as you have to think it your future. Okay, where you are, what you want to do, plan everything carefully. Then only you can reach the height. Okay, but you have to enjoy, obviously, because this is your age. You have to enjoy. But the concept is you have to. There is a target should be there. And then see, this is the addiction of the cell phone. And then don't adapt to this. Is as far as my concern, that's what I didn't bring my phone. So don't be as far as my concern, cell phone is an evil. Okay, don't go to back to future. Okay, then think outside the box. Think outside the box in the sense, maybe to identify yourself or to identify others. He's the person. So you should not. You have to think wisely. That may be funny to others. That maybe your concept may not be right, but there may be a spark in that if you go in deep. So that's what you think outside the box, right? Like Rajinikanth now. So why Balachandar chosen Rajinikanth? Okay. And then success. How many of you written the CSA exam last month? See, MSc second year. CSA எழுதலையாரும்? So you are giving the opportunity to the North Indian peoples. Please. Success is not the straight line. So your parent is well-to-do, that is a different story. Better to stand on your own leg. Okay. So the success is not a straight line. Obviously, this is the one. Okay, I think you may think I'm an expert or your teachers are expert. Obviously, anything was once, they are the beginners too. நான் இங்கே தான் உட்காந்துருந்தேன். right? Okay, I think okay, I think if you are really interested, even I remember what kind of seminar I attended when I was studying my MSc. The first lecture, who taken, what is the title of that one? Everything is in my mind. Okay, so you have to develop. If you want to stick with, yes, this is my career. This is I want to stick with the science. Those I'm advising only those people, not for others. Okay. So you take a filter, put it, the filtrate, who is the filtrate? Okay. So I think, please, that is the inspiration. Okay. Sorry, they are my PhD students. Maybe, Path sir. So they are my PhD students. I think most of them are in abroad. If I go to any country, I never pay for taxi. They'll come in and pick me up. They will drop me by their own car. You name the country, either Australia or New Zealand or Estonia. Okay, I think my students are there. I think I got many awards. That's what they told. நீங்கள் பண்ணுங்க இன்னும் ஒரு இது வந்து என்ன, this is our current group. They are my victims. Whatever I am talking to you, they only did it. I am simply putting my coat, and then I am talking to you because that is their hard work. Okay, okay, okay. I think this is a Christian college, so I am very happy to say I visited the holy place, Jerusalem, for many times, 4 or 5 times. I have collaboration in Hadassah Medical Center. Okay, so I think this is the Dead Sea. Dead Sea, you can if you enter into the Dead Sea, you can float. Okay, so this is one of my friend having book. That time I don't have nothing, so I borrowed the book and then I simply floated on the Dead Sea. Okay, next slide. So this is that Sea of Galilee, and yeah. All right. So I think I don't know where to stop. So my sincere advice is, please educate yourself. Okay, don't read only whatever the discipline you are studying right now. Extensively studied, like a study. Either the botany, zoology, forget about that now. Even if you want to talk about rocket science, you have to learn rocket science. Okay, this is the strong foundation you have to create yourself. Okay, so I think with this I will stop. If you have any question, concerned with I think is the beauty. They told about, Madam told about the beauty. Beauty, the beauty, as far as my concern, till the depth of the collagen in the skin. Right? உள்ளுக்குள்ள இருக்குல்ல, that is the real beauty. Right? The beauty is the depth of the skin. Okay, forget about it. I think there is a lot of comparison, like a young woman and a old woman hand together, So collagen, as far as a scientist, I am thinking there is no collagen. Okay, I think I will stop here, madam. Thank you.
A very interesting session by Dr. Sundaresan. Any questions? You are most welcome.
Sir, good afternoon, sir.
Okay, the respect, better to be in your heart, there's no point to because when I was in AMERICANCOLLEGE, I will call my boss by name, Sundaresan. If I ask madam, no, you have to call me Sundara, something like that. Please.
Sir, I am Sharon Rubini, Department of Mathematics, sir. I am working on single gene abnormality. You talked about single gene disorder, sir. Yes. Have you come across any patients here, sir, in Aravind? And did you have a treatment and diagnosis? What was you carried on?
There is a disease called aniridia. This aniridia. Aniridia in the sense, Aniridia in the sense absence of iris. No, iris is there. The iris, please recall the structure of the eye. Iris, the iris secrete the aqueous humor. The aqueous humor, it will go through the Semmes canal and then it will reach the episcleral vein. That is the flow. If there is any block in the path in the flow, it will affect it will increase the pressure and then in the optic nerve damage. Okay, the aniridia is an excellent question, madam. Is aniridia is a single gene, single disease. There is if the iris like a complete iris, if it is a complete iris is there, that is a normal vision. If it is partially, for some people it is partially there or only the rudiment. That is depending upon the effect of the thing. There is, please note down that the name of the gene is called PAX6. PAX6. That is the name of the gene. So that is one of the main gene responsible for the development of the eye. Now, during the development, this particular gene is playing an important role. Okay, PAX6. So that is a single gene, single disorder. Okay.
Did you come across some patients and did you have any treatment?
Madam, see, for your kind information, the egg and then the sperm, once it is fused, that's it. It means it is transmitted the signature to the zygote. Okay, so obviously that the signature is expressed to the patient or to the child. Okay, so here it is impossible to rectify after the zygote formation. It is impossible. You have to manipulate before that. That kind of technology is different. Okay, so here what we can do, how to avoid the transmission of that particular defect to the next generation. Okay, that is possible.
Yes, sir.
So what they have some additional treatment, like chemically. Those patients, they are photophobic. Again I tell you, they can't able to low vision, but can't able to see the bright light. So you can there is a lot of low vision aids are there. Okay. Through the low vision aid, we can go for the improvement program for the patients. But complete treatment, the rectifying it's not possible. Okay. But for some of the gene defect, like here, but they proved in the animal model, you just knock out that PAX6 and then you can generate the mouse. Those things, exactly it is mimicking as a human. Because all the experiments, they will do it in mouse model and then monkey and the human, So that is there, but there is no cure treatment for the patient. If we come to the back of the eye, that is called the posterior chamber, anterior chamber and posterior chamber of the eye. The posterior chamber, all the retinal defects, once the nervus is damaged, that's it. It is very difficult to regenerate. That is the problem.
Okay. Thank you, sir.
Yeah. Please.
So that's an eye-opening session, and thank you so much for this. So my question is about retinitis pigmentosa.
Okay.
So there have been that research is going on for past years, like still, and as far as I know, like there is no complete cure for retinosa pigmentosa.
Yeah.
But the thing is like. It may start from the teenage and it can go till 40, the complete loss that legally the person may be given as the blind as till 40 years.
Yeah.
So is there any thing that we can extend the age so that they can...
Okay, yeah. So I think she is talking about the Malakan Noi, okay? So the Malakan Noi, the retinitis pigmentosa, that is not a single gene, single disease. More than 65 genes are there for RP65. 65 genes. And then the mode of inheritance, autosomal dominant is there. Autosomal recessive is there. X-linked dominant is there. Recessive is there. Like that, because the candidate genes, as I told you, 60 genes, not only in a single chromosome, it is distributed in all the chromosomes. Okay? And then the severity of the disease is very high. As I told you, RP65, that the LCA treatment, there the retina is intact. The retina is intact. Even if you put the virus, there is no leakage in the retina for concern with the retinitis pigmentosa. So once it is they have the retinitis pigmentosa, there are lot of holes in the retina. Even if you put the retina, the virus for the treatment, it won't stay there for a long time. Okay, and then congenital stationary night blindness. There is one type. There are so many types are there. Autosomal dominant, rhodopsin. Okay, rhodopsin is the gene which is responsible for autosomal dominant disease. Okay, so there, but the frequency is low in India. In India, autosomal recessive and X-linked recessive is very common in India. Okay, so far, many clinical trials are going on in mouse model itself, but it is not in the human. That is the concern, So many people are, keep on asking, is there any improvement? Can we able to get the vision? See, for the cornea defect, at least they will get the hand movement. For this retinitis pigmentosa, I think again the diminished lux, that varies. They can't able to see. That is a major challenging, madam. People are, people are, doing many things. After that, they will write the cause is till unknown. You will end up with like that note. That's what we are, But we can understand the pathogenesis of the disease. We are a long way to go. I think you guys, maybe take the lead to give bring back the vision. That is you have to think differently how to cure this kind of diseases. Yeah, please. Hello. Everybody, இருக்கீங்க sir? Yeah. No, this is not. I don't know. My flow is not good today. Maybe I will can. I don't know. Yeah. Yeah. Till I'm mentally I'm young. Yes. Yeah. Okay. Okay. So I will speak in American language. So if I have a money, the money what I'm going to spend for gene therapy, I can get an island in Pacific Ocean. Okay, so for in the Valentine's Day, there is a heart-shaped islands are there in the Arabian Sea, that kind of thing, So like that, it is very expensive. That's what I put a word. If your Medicare, your insurance supports, definitely you can adopt this kind of thing. But lot of millions are there. I'll tell you for a kind information. In Portugal, and we got 1 million euro, from we got them 1 million euro. 1 million is how many? 10 lakhs, that much money as a donation for Aravind Eye Care System for a particular thing. That patient is affected by retinitis pigmentosa. Like Lady Tata, so he is affected by cancer. That's what, that family, I'm saluting him, they are supporting for cancer research. Lady Tata Memorial Fellowship, that is there is an opening. Please write CSR examination, Lady Tata Memorial Fellowship, One Child Fellowship. There are so many openings are there. Okay, please, explore, explore the possibilities by the government. You are paying, your father, mother is paying a lot of tax. Please bring them back money through you. Sir, that is the answer, sir. So you can get, if you have the money, you can get an island in Pacific Ocean. Yeah. In Africa, we have a branch. I think in Congo somewhere.
Is it one-time treatment? The gene therapy for eye?
No. Okay. Yeah. It's a very good question. So whatever I told you, the subretinal injection, in the retina. So maximum 2 injections. Maximum 2 injections so far they delivered. And then very recently, Jean Bennett, please, note down that she is becoming a millionaire because of this particular virus. One while, it's a huge amount of money. She sold her property, the company, to a different thing, she became a millionaire. I met her in Florida. Okay, so I think she is from Philadelphia. So they made the virus, clinical grade virus, that the virus is, we can able to inject. Okay, so, okay, thank you.
I think sir would have given you the right answer. We'll close down the session with the question from this side and one from the other side. Can be even in Tamil.
So break the barrier, no, just say something. சார் நீங்கள் மொக்கையே போட்டீங்க சார், என்ன சார் கொண்டீங்களா சார் அப்படின்னு சொல்ல கூட சொல்லலாம். Say something. So please, I mean, I think you enjoyed my talk. I can able to see the smiling face. You didn't sleep. Just, yeah. Yeah, I think it's very expensive. Yeah. So I think, in the Medicare in India now we are paying from our pocket. If see some people they can't able to diagnose if they have prostate. Fortunately, women not having the prostate cancer because the prostate gland only for men. After for some time 70-75 years, if you measure the PSA, prostate specific antigen in the blood, the level is high, they have the cancer. But 2 or 3 decades ago, even they can't able to diagnose whether it is a prostate or not. So now the scenario is different. At least we know the diagnosis. Obviously we are getting the computer bill. So if you have money, you can pay the computer bill. Otherwise, simply say hello to Jesus.
So if there are no questions, we'll close down the session. So let me close down with the quote by Theodore Roosevelt: "Keep your eyes on the stars and keep your feet grounded," which brings the message: aim high, have great ambitions, but do not forget to remain grounded and humble. So you can reach your ambitions, can be a great scientist, you can reach higher heights, but have your feet grounded. I am immensely grateful to Dr. Sundaresan, Senior Scientist, Head of the Department of Molecular Genetics, Aravind Medical Research Foundation, Aravind Eye Hospital, for his insights into the world of ophthalmic genetics. American College is a crown in Madurai and a pride of Tamil Nadu. Hope this endeavor will help in the development of science and technology. Thank you. Please assemble for group photo outside the hall.